I'm Ria Damani.
I'm Talia Simmons.
And I'm Savannah Wells.
So looking at all the different phases that we did, the first phase that we did was research. We needed a good base and a solid understanding to be able to continue on with the rest of our experiments. And also for phase 2, we did an experiment using baking soda and vinegar to replicate the cell growth and how quickly it would multiply to be able to see even further for our next phase.
For the third phase of our project, we did an experiment where we grew E. coli and we monitored the growth, and then we compared it to cancer cell growth. And finally, for our last phase, we'll be talking about the conclusion and what we learned. Moving on to the first phase, the research paper.
Okay, so here you can see some of the topics that we talked about in our research paper. We started with simply what were cancer cells. This was to get a better understanding of what cancer cells were, what caused them, why they were so deadly, things like that. And this set us up for the next couple phases of our project. Then we researched cancer cells growing out of control, why this happens, and again, why this is so deadly. And then we also researched common types of cancer to get a better understanding of where they form in the body. After that, we researched the deadliest types of cancer, specifically why more cancers travel fast into other parts of the body, while others tend to be more stagnant. Next, we researched how cancer cells are able to trick the immune system so our bodies aren't actually equipped with the tools to fight off cancer. And finally, we researched current and future treatments of cancer.
So some of the specifics of what we researched, so the first one is how cancer cells grow. This was obviously a large base for our future experiments that we did and also helped us set up our third phase very, very much.
The second big thing that we learned was what kind of treatments are currently being used, and this helps us set us up for phase 2, which we'll talk about in a little bit. When researching the deadliest types of cancer, this helped us understand how our experiments in phase 2 connected to our understanding of cancer, which we will cover in just 1 minute. Alright, so now we're going to talk about phase 2, which was our series of baking soda and vinegar experiments.
So for the baking soda and vinegar experiment, we used 3.55 grams of baking soda and 40 milliliters of distilled all-purpose and apple cider vinegar for each trial. As well, we used water and ice cubes as our changing factors, and we also used a baker's scale and a freezer to conduct the experiment.
So going into more of the specifics of what we did throughout the experiment, we had 4 different trials that we did, well not trials, 4 different methods that we used to be able to do the experiment. The first one being our control variable, and that was using 3.55 grams of baking soda and 3 different types of vinegars, apple cider vinegar, distilled vinegar, and all-purpose vinegar. And when we mixed the baking soda and the different types of vinegar to see which one had the largest reaction, which we then compared to how each cancer would react within the body. And then for the first treatment, we added water to the baking soda previous to adding the vinegar and saw if that reduced any sort of reaction.
And then for our third treatment, we put the baking soda in the freezer for 5 minutes and then reacted it with the vinegar to see if that would decrease the reaction at all. And then for our third one, we added ice cubes to the baking soda, stirred it up, and then added the vinegar to it again to see if that would lessen the reaction that occurred. Alright, so now we're going to talk about our results. For our control experiment, the largest reaction was definitely the apple cider vinegar. This experiment consistently bubbled out of the beaker. It also had the largest number of bubbles 1 minute after the reaction. Our smallest reaction is going to be our distilled vinegar. This experiment never bubbled out of the beaker. It also had the least number of bubbles 1 minute after the experiment. For our treatment 1, where we added 20 and 10 milliliters of water, this was definitely our odd one out, as it had no positive effect on the reaction. All the different types of vinegars all overflowed out of the beakers, and this occurred with both the 10 milliliter and 20 milliliter volumes of water.
For the second treatment, we tried chilling the baking soda. This was to represent a type of radiation therapy that is commonly used in cancer treatments. We found that it had the largest impact on the apple cider vinegar, found that it had the smallest reaction, and it also had the longest time for the reaction to reach the peak point. It also created a more foamy product compared to the others. It had the least impact on the distilled and the all-purpose vinegar. Most of the trials overflowed and were extremely quick to reach the peak height. For the fourth treatment, we added ice cubes to the experiment. It had the largest impact on the distilled and the all-purpose vinegar. It had a less reaction volume and a similar post-reaction appearance to our control group. And it also had the smallest reaction on our apple cider vinegar, and it overflowed most of the time. It was mostly bubbles and was slightly less foamy.
Okay, so we know it's kind of confusing as to how this connects to cancer cell growth. So we're gonna go over that and explain it more clearly. So we decided to use baking soda and vinegar because when we were planning out our experiment, we talked to Miss White, and she gave us the idea of using baking soda and vinegar to replicate how cancer cells would react in the body. Because once we drop in the vinegar, it has a very quick reaction and spreads like outward. And so then the different treatments that we did were to replicate like cancer treatments such as chemotherapy, radiation, and surgery. Alright, so now we're gonna have a live demonstration. So if we could have 6 volunteers come up here to help us.
So if anybody from the freshman class, the juniors, or the parents would be willing to help, as
Just head on
sophomores
up.
have
Seriously.
so far.
Just like come up. Whoever the first 6 people are, get to do it.
So that's 5. We need 1 more. Okay, we got 6.
Alright, awesome. So we're gonna have each of you grab a beaker. Alright, here.
We're gonna place this so we don't mess up the table.
Alright, each grab a beaker of baking soda. So here you guys go. And just hold on to that for now. Alright, so last person coming up. Alright, until then, so we're gonna have one person step forward. Alright, and you're gonna be the control group with distilled water. So seriously, just put it in and then just pour this in. So this is our distilled water. Alright, so next, just put that down. Alright, next we're gonna have somebody else come forward, and you are going to be doing
The control group for all-purpose vinegar.
So just pour that in once again. Alright, so now we're going to be doing our different treatments.
So for the first treatment, we're gonna take water, so 20 milliliters of water, and you're gonna add that into the baking soda first. And then now add all-purpose vinegar onto that.
Alright, awesome. So another person come forward. Alright, cool, so just put it in. Alright, then add the water. Okay, and then add the distilled vinegar. Alright, cool, so that was our first treatment. Let's do our second treatment.
So now for the next treatment, we are gonna put in 2 ice cubes. So if you wanna just put that in there. That's 2. Give this a little stir. And then pour in the all-purpose vinegar. We're just gonna move that to the side, and our last volunteer. Also gonna put in 2 ice cubes. Give that a stir as well, and then have you pour in the distilled vinegar.
Awesome, thank you guys. So I imagine it's probably hard to see for those in the back, but the treatments definitely did have an impact depending on the type of vinegar. And I know like the all-purpose and the distilled are like hard to tell the difference, but they do change how the reaction occurs. Just like how cancers can be similar, but they are impacted differently by the different treatments.
So now we're going to go on to our third phase, which was growing the E. coli. And the materials that we used, so we used a Luria Bertani agar plate, which is the plates down on the bottom, and we also refer to them as petri dishes. We also use inoculating loops to be able to transfer the E. coli cells to the agar plates. Obviously the E. coli cells to be able to start the growth, and an incubator to be able to maintain temperature.
Okay, so here you can see our results from our E. coli trials. We did 4 different trials. 2 were in an incubator with a controlled warmer temperature, and then 2 were just left at room temperature. So in the incubator, we had 2 trials. Both were around 37 degrees Celsius. And you can see that trial A grew a lot more than trial B, and we think this was partly due to like how we applied the E. coli, like it was more spread out in trial A. And then you can see from 72 hours to 96 hours, they didn't grow a lot, but they did get more dense in the areas that they had already grown up to. And then sort of same for the room temperature, you can see that they definitely didn't get as big as the ones in the incubator. And then between 72 hours and 96 hours, they did the same. They didn't grow outward, but they did get denser in the areas that they had already gone to.
Okay, so now we're going to be showing you what, if I can get there, what the E. coli actually looks like. Maybe it's gonna work.
Trigger. It's the trigger button.
Okay.
Just click outside of that. Oh, come on.
No.
Or I'll just go ahead up and change it.
Okay.
Why isn't that? You're getting very close.
So we're just gonna be showing you the trials that we did, and after 96 hours, we did put the E. coli in the fridge so it would stop growing. So what you're seeing is gonna be pretty similar to how it looked after 96 hours of growth.
Okay, so what you see here, these 2 petri dishes were the ones that were grown at room temperature, and they have been in the fridge for a little bit now, which stops growth, as discussed, but those were the ones that were grown at room temperature, and then comparing those to the ones that were in the incubator, we can see that those grew a lot more than the ones that were just out in the room temperature, as there was an increase of temperature in that.
Moving on, some experimental errors we had in our experiment was an inconsistent application of the E. coli onto the petri dishes. Most of the times when we applied it to the petri dishes, we just did it by hand and we didn't really measure the E. coli, mostly because we couldn't. So this could have caused for larger or smaller portions of the E. coli to be on the petri dishes, which would have affected the growth data. As well, the room temperature could have fluctuated, mostly because we left them in an incubator downstairs and we also left them overnight. For example, we left them over the weekend, and over the weekend the school could have shut down the power, and that would have caused a fluctuation in the temperature, whether it was warmer or colder, which also would have affected the data. And lastly, we had no real procedure during our third phase. It wasn't really strict at all, and we kind of just went at our own pace.
So now to go on to some of the changes that we would make next time if we were to do the experiment. So the first one we would make would be to use HeLa cells instead of E. coli. So HeLa cells are a type of cancers, or they're a type of human cells, and they can be infected with whichever sort of like drug or disease that maybe needed to be tested, and that would be able to work as one of the things we could do our treatments on, as that is not physically affecting a human, so that is something we would do in the future. We would also improve our treatments to be more accurate since currently there is no way to be able to fluctuate body temperature within one's body to be able to decrease or increase the amount of growth of these cells. So making our treatments more accurate to what they currently are and they will be in the future for cancer would have also helped, and we also could have done more E. coli trials overall, but we were unable to due to a lack of time and materials.
Alright, so I know that's a lot of information, so once again we're going to take a quick break to talk about how this relates to cancer. So, like E. coli and cancer are quite similar to each other. They both grow at exponentially fast rates, and they're both impacted greatly by the conditions that they're in. As you saw before, both E. coli and cancer grow better in warmer temperatures, but when it gets too hot, they're just not able to grow at a faster rate. Similarly, although we didn't do experiments relating to pH, both cancer and E. coli can grow in a wide range of pHs, but they grow better in different pHs. Alright, so our conclusion. So it was a really cool experiment, and we're all glad that we chose this. For phase 1, we talked a lot about our research, which helped us set up the rest of our experiments. Phase 2, we did a baking soda and vinegar experiment, which helped us understand how cancer grows and how different treatments affect different types of cancer. For phase 3, we did an E. coli experiment where we were able to closely understand how cancer reproduces within the body. The last thing we wanted to do was take a quick second to thank all the teachers and who helped us with this experiment, especially Miss Whitehead and Mr. Deedle. We couldn't have done this without you guys, and it was really kind of you to help us during this experiment. Alright, here are our references.
Thank you guys for listening, and do we have any questions?
Well, isn't there there's usually a range for E. coli to grow more comfortably, but I mean, it doesn't grow very well in colder climates, but there's not really much.
So if you basically put it in too cold of a climate or too warm of a climate, it usually grows best at the temperature we put it in the incubator at, which was, I believe,
like 37
degrees Celsius, because that's purely the temperature in the human body.
If you were to perform like a similar experiment, which type of virus and bacteria would you like to test?
I don't know. That's a really good question. I think it'd be cool to test like a variety, just to kind of see how they would react like in a controlled environment. But I can't think of one like I would want to do especially more than others.
Saba? What'd you say?
So the plates were already sterilized, and the inoculating tubes were also previously sterilized, and they were a single use, and then we had to dispose of them. So overall, it was pretty sterile.
Like we didn't make any of this stuff.
Right.
It all came from pre-packaged kits that we were able to change the variables, yes, but we didn't make any of the stuff. Yeah. Mr.
Why do you guys think the apple cider vinegar reacted more vigorously than the others?
That was partially the concentration of the actual acidity in the vinegar that caused a larger reaction with the baking soda compared to distilled and all-purpose.
It could also be slightly more acidic than the other ones, but we didn't measure the pHs because of the fact that with the baking soda, yeah, it could have been slightly more stronger that way. Charlie?
What was the most difficult part about the project?
I would say our last phase, just because it took with E. coli, because it took a while to set it up and change the variables. And there wasn't anything we could really do while waiting for it to grow. We just kind of had to wait for it to grow to a point that we could use. So I'd say that was hard just because we didn't really know what we were doing.
We also faced a few difficulties with the incubator just shutting off at random temperatures. If it was below I think it was 45 degrees Celsius, it would just shut off within 5 seconds. So obviously that first trial that we had to completely just remove. Mr. Deedle?
So in the experiment with the baking soda, the diluting the vinegar with water is one of the treatments,
Yes.
and then the ice is a second one?
Yeah, so the water actually went in with the baking soda, and then we switched it around, and then the vinegar went in after we added the water to the baking soda.
So are those just different conditions, or are they supposed to relate to specific cancer treatments?
So adding water is, it was kind of hard to replicate like actual treatments because they're very complex and we obviously didn't have those kind of resources. But adding water is supposed to represent like chemotherapy because it's like kind of lessening the reaction that would occur. And then putting the temperature in the freezer is like, it was supposed to represent radiation because radiation like kind of burns the cancer cells, so we just like froze them, I guess. And then we didn't know what the ice cubes were kind of representing. We just wanted to see if it would like dilute the reaction. Yes.
Okay, so, forgive me, but is radiation the same as chemotherapy?
No, they're a little bit different. Chemotherapy is like a cocktail of various different drugs that are artificially created. Radiation uses radiation to kill the cancer within the body. So like in terms of like lasers and stuff, that would fall into like radiation therapy. Yes.
What made you guys want to choose this topic?
So we had to do a book review for this class, like right before this project, and the book that I chose to read was about this topic, so that's when we decided to do this as our project.
I think initially we were discussing just being able to grow the actual cancer cells and being able to see like potential treatments actually being put into effect. That was definitely something that interested all of us, as we're all somewhat interested in the medical